Record on this. Okay. Well, I'm really happy today to be able to welcome Monica Berondo, who is one of the advisors on our project and one of the inspirations for a lot of the work we're doing. Um, she has her own company and does computational design of antibodies and wonderful stuff. Monica, I'll let you unmute and take it away.
Hey, thank you, and very happy to be here. And thanks again for accommodating me in my schedule. So what I want to do is break this up. Kind of half of this, I want to talk a little bit about my journey and my path of how I got here, and then half of it, I want to spend some time just kind of talking about general antibody engineering. I've tried to split it a little bit between how it's currently done and different methods for doing antibody engineering. And then kind of move that into computational antibody engineering, since that's kind of what we're talking about and a lot of what I've focused my company on. So I'll start with a little bit about my journey. I'm going to go kind of pretty far back. I was very interested in writing software, so computer programming at a very young age, and initially started in computer science. While I was in high school, I took classes at a local community college and just really enjoyed that. And I think that really kind of put me on that path. However, when I got to the point of actually having to work in computer science and doing some internships and some co-ops and spending some time at companies, the type of work I was doing wasn't very appealing to me. I did a lot of stuff on like writing front ends and back ends for websites and, doing some financial modeling and some software there. And it just wasn't as appealing to me. But I also happened to be working on a minor in chemistry. And so I was really lucky in that as I was talking to some of my advisors on the chemistry side, and talking about kind of some of the struggles I was having, they said, well, you seem to really like chemistry. Why don't you look into some ways of merging the two? There's a lot going on in this space. Computer-aided drug design was really big at the time. And there are a lot of people looking at how to do this. And they connected me with a professor at the university who was doing some modeling of proteins. And so I got to work in that lab really early on and just fell in love with it. I thought it was the most absolutely interesting thing I could do. I loved that work, and my advisor was super supportive and really kind of drove a lot of the decisions I went on to make. So as I was talking to him, I was like, I hate my computer science degree. I don't want to continue in this. What do you think I should do? And he said, well, I think if you really are looking for something, you should look for a program that matches what you're looking at. And he's like, biomedical engineering is kind of in that space and would be a really good match for you. But unfortunately, there are only a couple biomedical engineering programs that have that focus. So he helped me look for what schools had those programs and where I could actually work in that space and do my training there and kind of encouraged me to transfer to a different university where I could do that and really focus on what I was doing. And so that was really instrumental in getting me started in this space. So I transferred schools into a biomedical engineering program. I did a lot of my research in the protein folding and protein modeling space. That was most of my training was really on the research side, not so much in the classes I took. In the classes, since it was a biomedical engineering program, it was very broad. So I did a lot of lab work. I did a lot of tissue engineering. I did a lot of like devices, took some business classes. So it was pretty broad. But really that research I did is what drove me towards what I wanted to do. And so towards the end of that, I started looking into grad schools. I wanted to continue to study that. And so I looked at what programs were doing research in this. And they're kind of broad because, the protein modeling space tends to live in a very large diversity of programs. So you have biophysics programs, you have biochemistry programs, you have straight physics programs. Like you just never know. It's really more about the researcher and what they're doing. I knew I wanted to stay in engineering, so I focused on programs that were in engineering schools. So there was a lot in chemical and biomedical engineering, so that's kind of where I ended up. And so the way I went about applying to grad school was a little bit different, because I knew that the lab I ended up in was going to really drive the research I did, and I had a very focused idea of what I wanted to do. So what I did was I emailed every professor I could find that was doing this and asked them, is there room in your program? Can you find out from the dean or from the head of the department if there's room in your program still for someone who would work in your lab? Because oftentimes, not only do you have to match lab, but you have to match a program. And so that's how I ended up finding, you know, the handful of programs that I could, that I knew I would be able to match into the lab I wanted to match into. And I ended up at Johns Hopkins University. And so while I was there, I mean, I think that my choice ended up being just so ideal for what I was doing, because I ended up in a lab that was one of the labs working on Rosetta. So I'm sure it's pretty well known. So I'm sure a lot of you have heard of the Rosetta software modeling platform. And so I was in one of the labs that worked on that. And I was not only lucky enough to be in a lab working on Rosetta, but the timing was really great, because Rosetta was undergoing this essentially reformatting of the software. So they wanted to move the software from a very Fortran-based writing of the software into a more C-based, something that more people would be able to actually contribute to. And so I got really involved, not just in the research for my own research projects, but I got really involved in the development of the software itself. And I worked as one of the core developers to do that restructuring and that reformatting. So I got to learn about not just the aspects of application, which was what I was interested in doing eventually, but I also got to see how the innards of the modeling platform worked. And I got to work on pretty much all aspects of the software. And I think that really is what made it so that I was able to eventually start a company, is because I had been exposed to so much of the software that I understood how all of it worked and how all the pieces fit together. So I knew I'd known for a long time I wanted to eventually start a company. I just didn't know when or why or how or what I wanted to do that company for. And so I started talking to other postdocs and grad students that were working in Rosetta about it. And we were kind of coming up with an idea of how we could do something with Rosetta. And so we formed this group that was really kind of a semi-consulting group, where we would go out and train biotech companies and pharma companies on how to use Rosetta. And that's where I started to learn where was the idea of what I wanted to do. And so we did a lot of these training sessions. And one of the things that I found is that most pharma companies didn't have a good department for using these types of software. So they'd have one or two people that were working on supporting this ginormous system. And so we would go back and we would retrain these companies, because you know that one or two people would leave and you were left without a department or without that information on how to use it. And there was just really no good documentation or anything around that. And so that's what gave me the idea for starting Macromoltek, was that the software that was out there did not meet the needs of pharma, and the way that pharma worked couldn't capitalize on the software that was out there. And so that when I started Macromoltek, the idea was to first focus on antibodies, because it was 90% of what I was doing, and nobody wanted to touch antibodies because they're like, oh, it's such a boring academic problem. It's not really anything super interesting. Whereas pharma is like, everything we do is antibodies. We need antibodies. We need better ways of developing antibodies. So that was the first thing. And the other thing is just to work really closely with biotech and pharma companies to make sure that we were functioning in a way that fit their system and their needs. And so the initial idea around that was to do a software as a service. So we thought, you know, everybody's moving to the cloud. What if we set up this, like, instead of having installs of software at companies, what if we had this like cloud-based portal where everyone could just do whatever they needed to do. So you could do antibody modeling, you could start to do design work, you could do different things. You wouldn't have to download any software. But the business model around that is very tricky, and pharma companies just weren't set up to do that. And so they kept saying, oh, this is really cool, we love your portal, why don't you just do this for us? And we started turning into more of a consulting firm, because they would just send us stuff, we would do it ourselves, and then we would just send them back the results. And that worked great. Like, that was amazing. I got to learn so much. I got to work with amazing people at different companies. But the business model of it wasn't really what I was looking for. Like, I wanted something that could expand, that you could grow very well. And consulting isn't very easy to grow into something big. And so around that time, we got invited by, I think it was a researcher at Johnson & Johnson, wanted to bring together all the different software platforms, be it academic or industry, that were working on antibody modeling. So how can you build a better model of an antibody and give them a blind assessment? So if you've heard of CASP, which is kind of a blind modeling of protein structure prediction, it was very similar to that, but very focused on antibodies. And it was really awesome, because we got invited, we got some articles out of it and some recognition of what we were able to do. And we were the only company that was like really focused on antibodies, that was working on it. And I think that really helped us get a lot of companies interested in what we were doing. And so one of those companies was a local company in Austin. So I'm in Austin, Texas, and it was a government contracting firm. So this is a company that all they do is go and work with government contracts. So this could be something like a DARPA, or there are a bunch of other things similar to DARPA, or it could be an SBIR, but everything they did were these government contracts. And that's how they worked with industry. And so we merged up with them because they got this really interesting contract where what they had to do was build antibodies. It was rapid development of antibodies. And so they had heard about us through this assessment and said, well, can you use what you're doing and apply it to designing antibodies? And we're like, well, yeah, I mean, we have enough information about how antibodies worked. We've done a lot of things like optimizing antibodies at that point. And so I said, sure, we can try to do that. And since it's under a government contract, they want like just really super innovative ideas that are not necessarily guaranteed to work. So it was a really good way for us to test something we had never tested before. And that turned out to work really well, because we were able to design an antibody completely from scratch. They did all the lab testing for us. And so that was really great, because it helped us focus in on a way that we could develop a different business model. Up until that point, we had, like I said, we'd done a lot of work with consulting, but we had also gotten a couple of grants from the National Science Foundation. And one of those grants was for taking what we had done in modeling antibodies and applying it so that we could do automated antibody humanization. So humanizing an antibody is where you take an antibody that has been discovered normally in a mouse, but it could be a rabbit, it could be a goat, it could be anything, any animal system. And you can't take that and use it as a therapeutic, because it's just not going to have a good reaction in a human body. And this is a process that's been done for years and years, but most companies have their own manual process. So it's a combination of know-how and picking by eye and by hand how you want to change the antibody. And we're like, you know what, everybody does it differently. What if we could come up with a method that, regardless of what antibody you fed in, always gave you the best human antibody out? And this was really important, because one of the big problems is that since people were doing this differently and kind of like by eye, most of the time the antibodies that you got out, while they looked very human, just didn't work. So it was common that you would get a 50 to 100% loss of activity once you humanized an antibody. And so we worked with a collaborator here at the University of Texas who had a really big interest in humanizing some of their antibodies. And we thought, okay, well, if we merge together and work on this grant together, we can set up something that's automated, test it on just a bunch of different types of antibodies, and show that no matter what we put in, we always get an antibody that works within 10% of the function of the antibody you put in that was not human, but looks as human as possible. And so it was the combination of these two pieces, this humanization that we had done, and this subcontract we had done, where we showed that we could design an antibody that really switched the direction of the company. So around that time, we're thinking, okay, well, how are we going to change our business model? Right now, we look like a consulting company. We still have this SaaS thing that we keep trying to get people to use, but it's not the business model we want. Right now, we look like a software company or a consulting firm. We want to look like a biotech company. So how do we make that change? And so I spent a lot of time talking to like advisors and software companies and biotech companies to try to figure that out. And at the same time, a friend of mine said, hey, you should apply to Y Combinator. And I was like, yeah, I don't know. They don't really do biotech that much. But whatever, many conversations later, I was convinced that it was a good fit. And so we applied to Y Combinator and got in. And my focus there was really just this question of business model, is how do we take what we're doing now and change it into a business model that looks more like a biotech and that still works with biotech and pharma companies. And that was our primary focus through Y Combinator. And so we went through Y Combinator, and it was really great because like I said, we focused on changing our business model. So how can we capitalize this? And we changed from something that was a pure consulting model to something where we could start to actually generate value, something that looks valuable to pharma. So when you're taking an antibody that a company gives you and making a small change to it, it's very hard to convince them that you're generating new intellectual property to them that is unlike something they could get otherwise. But when you're designing a completely new antibody, now you're generating IP out of nowhere, right? And so that is something that you can convince them is they couldn't have gotten without you. And so that's what allowed us to change our business model so that we really look like a biotech. So someone comes to us, they give us a target of interest, and we design an antibody, and now anything they do with that antibody, we essentially own a part of it, or if they own all the rights, they owe us anything that they make off of that. They owe us some percentage back. And so that really was a big change in how we worked. And again, like that was a really big change. We'd gone from being like a 2 to 3, maybe 4 person company to actually having to grow this into something that can actually grow. And so the first thing was we needed money. So with this new business model, our timelines kind of went out, because when you're consulting, these are small projects that you get money just, and it's very reliable how much you're going to get. But with this new business model, we needed some room to be able to say, oh, it's going to take us a year to do something. And if we're not making money in that year, we can still, afford to be alive. And so the first thing we did was raise some capital. The next thing we did was hire a team around that. So if I was going to be doing more of this business development side of it and fundraising, I needed a team that could actually execute on these designs. So we built out a team around that. And then the third thing is that if people were going to believe that our antibodies worked, we needed to be able to take our designs that we had made computationally and actually test them. So we had to build out a lab. So that was the third component. And so that was really the majority of what we did with that raise. And that allowed us to actually grow out our business development and the number of projects we had to really just maintain the way we are now. So I'm going to kind of wrap that up. It's obviously still an ongoing story, but that's kind of gets us to about modern day. And I'm going to move more towards the design side, but I don't know if it would make sense to take up a couple questions here or just wait until the end. I'm happy to do it.
Anybody want to ask questions right now?
Jump in.
Sure.
Monica, are you using artificial intelligence in any of your design?
Yes, and I'll cover that in the second half.
I have a quick question. So how big is your company now?
It kind of fluctuates between like 15, 20 people. Depends. We do a lot with like interns, and sometimes we have a lot of projects. So we kind of bring on people that are part-time for that.
And you've had interns from Austin Community College, am I right?
Yes, we have. Mostly in our lab, so in the actual building of the work.
So what's the distribution? Like how many lab people, how many other people?
Yeah, it's still mostly computational, and we do outsource a lot of the wet lab. So it's really just that validation piece that we do in-house. If we have to do like cell-based assays, or if we have to work with mammalian cells, we outsource that. So it's about 4 people in the lab, and then the rest are all computational.
Thanks.
So do you have a mixture of consulting as well as making it more of a business, or have you completely shifted over?
I'd say we're about 90% shifted. We do occasionally still get interesting enough projects that we'll do consulting, but it's very hard because it's just not a great business model, because it's not very reliable.
Oh,
Okay.
Shisha.
Monica, I heard you mention wet lab. Are you doing recombinant engineering to produce the final product?
Yeah, so we do, if we do it in-house, we produce SCFEs, or the heavy chain variable region attached to the light chain variable region. And we do those in E. coli. And then if we're producing monoclonal, then we outsource that, and that's mammalian production and recombinantly of the antibodies.
You said Goli, is that the cell line?
Sorry?
I heard you say Goli.
No, she means E. coli.
E. coli. We use
E. coli. I'm sorry. Thank you. All right.
Okay, one more question, and then we'll let you continue. Shisha.
Yes, hey Monica, I'm Shisha Huang from Baylor College of Medicine. So I run a core antibody-based protein course. I use a lot of antibodies.
Mm-hmm.
So we buy antibodies from company. We like CSV antibody the most, because that's the one can validate most often. So I was wondering, for the antibodies you design, do you have the idea of very specific to this particular protein in mind, and are those antibodies commercially available? Can we buy that?
Okay, so if I understand correctly, you want to know if our antibodies are commercially available, and then a little bit more about what types of antibodies we make.
And
Yeah, so no, I mean, we don't produce antibodies to sell the antibodies themselves. We produce IP that somebody buys and then sells. So we work with like reagent companies, and we design the antibodies for them, and then they would put them in their catalog.
Okay. So
and then I think, go ahead.
Yeah. So when you say you design the antibody, so you design the region how to produce the antibody. You mentioned the molecular antibody production. So you still need a lab to produce the antibody.
Right. So we just use very small quantities of them, just enough to show that they work. So we do validation ourselves, and then either we work with CROs to do. We mostly work in therapeutic antibodies. So we work with CROs to actually do all of the drug development side of it, or we partner with a company if it's an antibody that would be used as reagent, and we sell the IP to them so that they can produce the antibody and sell it on the market.
Thank you. This is incredible.
Sure. And then I think I heard another question kind of in there. And so the other thing, so what we're doing is designing the variable region of the antibody, and we're designing really just that binding. And so I'll get into this in a little bit. I tried to keep it pretty high level and not just focused on what we do. So if you have questions more specifically on us, I can do that. But really what we're trying to do is find an epitope that will have whatever the desired response is. And once we know that epitope, then we're designing so that we get like kind of a click into that and get binding directly to that epitope.
Monica, we have one more question.
Sure.
and that's from Sheila, who is asking where you learned some of these skills.
Where I learned some of which skills, because there are many skills. So on the lab stuff, I think it's really just the undergrad work I did, and I really don't do much in the lab. Like I just hire really great people to run our labs, because my lab work is so far back that I wouldn't consider myself to have skills in the lab. All the computational stuff was, I mean, growing up, like I said, I love working on computers. Everything else has been from the research I've done, or I've picked it up along the way. A lot of what we do, like I've invented a lot of the algorithms that we use and everything. And then the business side of things and the managing of the company, that's really been on the job. Like I don't have any formal training. It's been from working with really great people. So like for business development, just hiring someone who knows how to do that. How do you write a contract? You know, you hire someone, they kind of walk you through it a couple times, and then you can do it yourself. But yeah, like the business side has definitely been a very steep learning curve for me.
All right, we'll let you get back to it.
Okay, so for this part, I do have some slides, and it's a little bit long, so I'm going to go through it kind of quickly, but there are a couple things I want to cover here. Let me just share this real quick. Okay, I think that's sharing the right screen. You should see a big blue screen that says Macromoltek. Yes, okay, great. Okay, so I'm going to start kind of basic with, you know, why do we care about antibodies? Why do different companies care about antibodies? And the main thing is that these biomolecules are bodies already produce that are able to fight specific diseases. So they already have these really amazing drug-like properties, and it makes it much more seamless to integrate into our natural defense mechanisms, rather than doing something like a small molecule where you really have to kind of fight with your body's environment in order to make it work. The other thing that I don't talk about as much, but that I want to point out here, I think, especially given the question Shusha had, which was that these are very specific molecules, so they make amazing reagents. Most of the work you do in the lab, if you want to be able to detect something, is done with antibodies, and that's really based on the specificity. Since most of what we focus on is therapeutics, this is really driven towards therapeutics, but a lot of the same things apply when you're talking about diagnostics or reagents or just using antibodies in the lab. And so compared to traditional small molecule as drugs, antibody therapeutics can act on what have previously been considered undruggable targets. This is because of their large size, and they have a much larger surface area with which they can interact, which makes it so that they can target things much more specifically. In addition to this, since they target things more specifically, you can cause fewer side effects. So it's a lot easier for your body to deal with these than with small molecules or other types of molecules. And this is really, you can see this in the clinical success rate, where antibodies are twice as likely to succeed as a small molecule counterpart. So, before I get into the computational side, how are antibodies made today? And so this comes from two main sources. So either using the immune system of an animal to generate an antibody, or engineering a microorganism. So the most common engineered microorganism would be phage, and using phage display to produce these antibodies. For both, you take your target of interest, you prepare it, and you present it to the platform. So you either inject it into an animal, or you put it in the mixture with your phage, and that's what allows these systems to produce antibodies. In animal immunization, the animal's immune cells produce the antibodies and attack this disease. And then in the display technologies, microorganisms are engineered to display antibodies on the surface. So they're kind of like on the surface, and then it'll bind to it, and everything that doesn't bind drops out. Some of these bind to the target, and then you still have to filter through. And so one of the biggest problems is that you have to filter through a ton of antibodies in order to find the one of interest. And so animal immunizations can generate really great leads, but it's really time, labor, and capital intensive. And like I mentioned with the humanization work that we did before, animal antibodies are recognized as foreign particles still. So you have to either engineer the system to produce the antibodies that are human already. So these are like engineered mice, or you have to take the antibodies that come out of the system and then engineer them with something like a humanization to get there. And this is really the reason that display technologies were even invented, was to address some of these problems. And so there are a number of different display technology companies that give promising antibodies against any target. but they can get you these leads, but these leads just don't look like natural antibodies. And so they have a lot of manufacturing issues, and they tend to die out in preclinical testing. So despite 3 decades in development of these phage or display antibodies, very few of them have gone on to clinical trials. So the bulk of clinical stage antibodies are still coming from animal immunizations. And then one of the other critical flaws of these platforms is that they just can't target a specific mechanism of action. So this is back to that question of like, how are you targeting it and what's different about using computational systems? And that's that you can focus in on where you want your antibody to bind instead of just having to set up so many experiments in order to be able to find those antibodies that have the right mechanism of action. And so, and then there's this whole development pipeline that happens after this. So the thing that we want to do, and the reason why I get really excited about computational methods, is that you want to change it from a discovery process into a design process. So this is really like comes from my engineering background. Like I really want to design a system rather than relying on just picking through things to find what works. And so I'm going to kind of give a brief background of like the different parts of the antibody, because that'll help. I know I've already kind of talked about some of these things, but you have your antibody, you have the light chain here and the heavy chain here. The binding region, which is where we focus, is up here. And then this is called the variable region. And really specifically what we're working on is this paratope, or the binding loops that work on an antibody. And that's what's able to really pick up really unique different epitopes and go after different targets so specifically. I'm gonna skip this since I already kind of talked about it, but again, like using immunization or other methods is just really slow and takes a lot of work. And then I have this joke I like to put in here, and I hope you will appreciate it. But so Moore's law states that the computational power doubles every 2 years. But Mouse law does not state that the computational power of processing power of mice doubles every 2 years. So this really just kind of gets you into that mindset of like, no matter how much engineering you do to mice, you're really still going to be with this set system. Whereas if you can take advantage of Moore's law, and you can take advantage of the increase in processing power that we've seen over past 5, 10, 20 years, you can do things that you wouldn't be able to imagine otherwise. And so this really moves everything from like a lab-based system to a cloud-based system where you can do things a lot easier. And you can see this if you look at kind of the development timelines of using a traditional hybridoma, which takes 6 to 12 months to get an antibody hit, versus if you do this ab initio. So you might not get your final hit here, but you can do this so many times in the same amount of time that the likelihood of you finding that really specific binder is so much higher. Okay, so now I'm going to really move into the computational side of things. So why computational? What are some of the advantages? So the first is that you can go after inaccessible epitopes. So since you can take a protein and you can manipulate it and find different epitopes in the protein, you can go after areas that might not be found in nature at a high enough time scale to where a mouse is going to be able to produce antibodies against it. And then you can isolate the system, so you don't have to worry about, is there something else that your antibody is competing against to get to that specific epitope that you want to target. Then the other thing you can do that's really cool is you don't have to rely on that humanization process, because you can design antibodies directly on human scaffolds. And that allows you to get antibodies that are kind of like further along the process than they would be if they came out of another system. Okay, so just to show you kind of what this binding region looks like when you're looking at it on a structure. Again, this would be this Fab region. So these two pieces you see up here in the structural way that we look at antibodies. And then you have your variable region would be kind of the top half. And then this like different color, the yellows and greens here would be these loops that bind to, or the paratope that binds to your target of interest. And then, so I'm going to talk really broadly about antibody design and like the different modes. So there are, most existing methods of working on antibody antigen complexes rely on just the chemistry behind it. Whereas ab initio would mean that you don't have any information in advance of this. So you don't have a starting point for an antibody, you don't have existing chemistry, you kind of have to come up with that from nothing. And so, an existing or a development or non-de novo method would take existing antibody, make a series of individual amino acid mutations, see how that changes this interaction energy, and then focus on just improving or changing an existing antibody. I think that's the same slide, sorry about that. The holy grail of this, of computational antibody design is, if you just have information about the target, can you design something from scratch that will work with the mechanism of action that you're looking for? And so this means that you have a really low information requirement because you only need an antigen structure, but there's a very wide space that you have to search to get there. Sorry, I had some duplicate slides in there. And so what we do in order to be able to, or what you try to do in order to get there is work on a couple different aspects of that geometry. So the first is the geometric. So can you find a shape that looks like it would be opposing that region? So does your paratope look like the opposite of your epitope? Then you look at the chemistry. So what are the interactions that drive that, the binding? And then you kind of iterate over those different things until you find something that fits. So a little bit more about the biochemistry. Really, there are a number of things that represent that. So the first is hydrogen bonding. So this is, kind of like a seatbelt coming together. Then there are charge interactions, which are like two magnets coming together. And then the other one, which is not quite the biochemistry, but more on the shape side, is the shape complementarity. So this is what I was talking about. Like, do you find an opposing shape on the paratope to the epitope? And so this is all really great if you're looking at it in terms of structures, but what if you want to do this on a large scale? And what if you want to start to take advantage of not just biophysical methods, but AI and machine learning techniques? Then you have to change the representation of these features and turn them into something that the computer would understand. And so that's what you do here. You take an antibody or an antibody and an antigen, and you represent it in one of two ways. So the first way is to represent it as a graph. So this would be a graph of interactions where you say you have, little balls or nodes where your amino acid would be, and edges that represent the type of interaction between that. So you could have a network of two graphs where you have a graph for your antigen, a graph for your antibody, and then a different graph for that interaction. So instead of being bonded, these are not quite bonded interactions between your epitope and your paratope. And then the other way to represent it would be using voxels, where you kind of make little boxes around regions. And you change the size of those boxes depending on the type of interaction that you want to represent. And this really starts to get you to where you can take advantage of a lot of the computational power behind things like GPU processing or machine learning. Okay, and then when we talk about computational design, there are kind of two main questions that we have to address. One is, where on the antigen do you want to bind? And the other is, which of the designs work? And so what we do is represent these in two different ways, one with epitope mapping, and then the other with antibody selection. Okay, so I'm going to get a little bit into the process now. So the first thing is, you have to understand that without a lab, it's impossible to validate whether your antibody works. So a lab is very important in order to be able to take these antibodies, make them, and then go back and make changes. And the really great thing about computer systems is that you can teach computers. So you can take the data once you test something, show it to the computer, and the computer can learn from that and know what not to do next time. And so that's kind of what I'm representing here with this circle that, you know, kind of continuously flows. So say you have, you know, an antigen you want to design an antibody against, you would generate a ton of different antibodies, run it through some neural network, and that neural network will tell you, these are the antibodies we think will work. You test that in the lab, and then you take that back, the response back, test new ones based on those antibodies, and you get better and better every time. And so the nice thing about this process is that while I showed you that with like hybridoma, it could take 9 months for a single cycle, something like this you can get to take a couple of weeks, depending on your antibody, the way that you're actually producing those antibodies. You can get that down to be very fast. If you're doing mammalian expression, then you're really just constrained by the expression time. And so this is kind of the basis around how we can improve things and really learn from it. And so this is a little bit about our process. There are many ways that you can do this, but our process has 3 primary steps. The first is to take our target antigen, turn it into that graph. So again, I told you you can represent these different interactions by a graph. The nice thing is that with a graph, you can look for different features that you wouldn't be able to find it by yourself. So the computer can find these features. And with this, we can get probability maps that tell us where an antibody could bind. And this is what we call our epitopes. Once we know that we have these epitopes, we have these regions which we can design the antibodies against. Then the next step is to just run an algorithm that's going to design antibodies. And the important thing here is to keep in mind those main features at the interface that drive interaction, and that's that shape complementarity, and the biochemistry of interaction. And so there are many ways that you can do this. We have our own, which we think is really amazing and samples this area very well, but there are a lot of different ways that you can do that. And then this will give you a pool of antibodies. And then the next thing that you have to do is figure out which one of these antibodies is the right antibody, which ones are the ones that are more likely to actually interact with your antigen. And so, you know, more traditionally, this has always been done using an energy function from some sort of a modeling system. The way we do it, and I think what makes this really unique, is that we use neural networks for this. So again, you can represent that area of interaction with a neural network, and with those graphs that look at those interactions, and then you can continually teach that neural network what works and what doesn't work. And this allows you to actually pick these antibodies and come up with antibodies that are more likely to bind. And yeah, with that, I think that's all I have. I wanted to make sure I saved some time for questions, since I'm sure there are a lot out there. I will stop sharing, but I can always bring up my slides again. Yeah, so I'm happy to answer any questions more specifically about any of these different pieces, or about the whole process as a whole.
It looks like Shisha has a question.
Go ahead.
Yes, thanks, Sandra. Monica, this is fascinating. So you teach the neural network what's specific, what's not. During the process, did you mention that you also have to have a lab to validate those antibodies?
Yes, exactly.
How does that work?
So yeah, I didn't talk about the lab component as much, but I can go to really briefly. So once we have a sequence, we take that sequence and express it. So like I said, we can either do SCFBs in E. coli, or there are other ways that you can produce the SCFBs, or you can use a mammalian cell line to produce them as a mAb. What we're looking for there is just the recombinant antibody, and then we take the recombinant protein or antigen of interest, and then we do SPR to figure out what is the binding energy between those. So
okay, you just produce
the SC what. SCFV, so it's a small chain variable, no, fragment variable. It's single chain. So it's taking, normally you have your heavy chain and your light chain, which are two different proteins. And so what we want is a single protein. So you take that heavy chain, then you put a linker, which doesn't affect any of the mechanism or the function of the antibody. So you put a linker to the heavy or the light chain, and then you now have one single chain, which makes it a lot easier to express because it's like expressing a single protein versus two proteins and then making sure that they come together.
Okay, thanks. So just quickly, a quick follow up on that. So with that synthesized antibody, I mean, it's at the final stage of production to being used. Is that sufficient, or do you still need to produce?
Right.
Another
So
way?
it depends on how you're going to use it. So we make antibodies for a number of different functions. So if what you want as your final output is an SCFV, then yeah, that's sufficient. A lot of times you want an SCFV if you're putting it on a bispecific and it's like the SCFV attached to some other monoclonal, or if you want to use it in a CAR T, you need an SCFV anyway. So it's just the SCFV. Most of the time, however, it does get reformatted into a monoclonal. And so then it would go into another production system.
Okay, thank you so much. So my lab does large scale screening on antibodies or on other things. I think maybe you are doing large scale screening platform as well.
I'm not sure. What do you mean large scale? So our wet lab is really just validating the antibodies that we design. So we're not screening antibodies.
Oh, okay, sounds great. Thank you.
Screening is all done computationally.
Okay, thank you.
Yeah.
Thank you, Monica, very much for a great talk. So I have a question. How do you pick the original epitopes? Because the functionality of the final product, irrespective of what format you have it in, is dependent upon where they bind. Or do you just make antibodies to every surface you possibly can?
Yeah, so that's a little bit of our secret, but part of it is there are two components. There's where can an antibody bind, so that you can do computationally because you can use computational ways to figure out like what regions look like something where an antibody can bind. But then a lot of what's behind making sure that it's functional means that you're doing a lot of bioinformatics to figure out either does it look like a region that's something else, or do you understand the biology of the protein well enough? So there's a lot more that goes into figuring out where to bind an antibody, and it also depends on what is the outcome you want from it. So sometimes the outcome means that you don't really care as much about where exactly it's binding. You just care that it's avoiding someplace, or you care that that it's blocking an interaction. So we take all of that information and kind of bring it together to figure out where to target.
So you really have to understand the surface of the
Correct.
No, I am just the surface of the protein and the functionality. Like for CAR Ts, you want them to bind closer to the cell membrane than away from.
Exactly. Yeah.
Okay. Thank you.
Okay, I'm going to ask a question from the chat and then I'll get to Aaron and Daniel. So Keith was asking about the size of your neural network and if your computational power is in-house or outsourced.
I'll answer the second one because it's an easier question. So we have our own internal servers. The size of the neural network, we use so many different ones. I don't know that there's like an answer to that question. It also is our proprietary algorithm, so I couldn't tell you too much. But we have a combination of some shallow and some deep neural networks that we use. We've built it off of both simulated and experimental data, and then also our own experimental data that we add to that. So it's kind of a combination of a bunch of different neural networks coming together.
He also had, if you had a metric like a hit or miss ratio.
Yes, so we consider binding below 100 nanomolar as measured by SPR. I saw someone ask what SPR was. So SPR is surface plasma resonance, and it measures the interaction between your antibody and your antigen. So it's giving you a value of how tightly they bind, how tightly the binding on rate and off rate is. And so we use the KD, or the combined on off rate. And that's what we use as our metric. So like I said, less than 100 nanomolar. And if you ask many different people, everyone has a different yes, no answer of what they want. If you're talking to a pharma, they want it to be picomolar. If you're talking to another pharma, maybe they want it to be 150 nanomolar. So it's very all over the place, but we find that under 100 is something that's doable by computational methods. And it's also something you can improve upon very easily.
Aaron.
Hi, this is perhaps more of a naive question from someone who know very little about antibody in the field. So my question is, I know several people that I know of in the industry work on the scFv, because as you mentioned, you can map it back to the monoclonal or bispecific or something like that. But at the same time, I also know some people also, I've seen people working with the nanobodies. So my question is, what is the current state of the use of nanobodies in perhaps therapeutic or any biotech diagnostic or something? What's the relevance, I guess? Because I know the size is small. The argument is you can penetrate better for certain in area and all that. But again, I have yet to see the actual application in the sort of like the therapeutics, right? So.
Yeah, I mean, I think it's a pretty new field, so there's not that, I mean, everybody is interested in seeing what they can do. I don't think there's a good answer for how it'll be used, because different companies have different reasons why they would want an antibody. Sometimes you can get something that's more specific. Sometimes you really want something that's smaller. So the whys are various depending on who you're talking to. The difficulty really is in getting not just nanobodies, but unique and specific nanobodies. And so the problem right now is that there aren't very many companies that can even generate nanobodies or do discovery of nanobodies. You can only do it in something like a llama. I mean, llama is pretty much the most commonly used to get nanobodies. And for computational methods, the data space is very small. So it's a little bit harder to get algorithms working as well on that as you can with an antibody. So it's definitely a space that's under a lot of development, because they're very, very hot. You know, a lot of people really want to get nanobodies.
I see. So I have one more. Thank you. I have one more question actually. So the small size, will that make an impact in terms of the binding? Because the reason why I ask is because some people use the antibody as a delivery vehicle. They can conjugate it to some nanoparticle or whatever, right? But nanobodies considerably much smaller. I think 10% of the mass, the antibody, right? So will attach this this big stuff will affect its binding to the target?
Yes, so when you have a nanobody, the primary thing for binding is that you lose the light chain. So it really depends on the epitope and what the epitope looks like, whether that loss of the light chain is important or not. So you have, if you look at antibody-antigen interactions, you have antibody interactions where it's primarily just driven by the heavy chain, in which case losing the light chain is not going to have a huge effect on the binding. But then you do have some where it's like this really large surface area, and where that loss could be very detrimental. And so you have to find a way to fill that in. It also depends on like the size of the epitope you're looking at. So nanobodies are oftentimes looked at for really areas where you have to get into something very small, because you don't have to have that extra light chain in there.
Thank you. Thank you.
Okay, Daniel.
I think Todd was ahead of me, actually. He raised his hand first.
Oh, go ahead. Go ahead. Yeah.
Oh, okay. I was just curious, and again, I'm kind of naive about this as well, but you were talking about 3D shapes fitting each other as part of your approach. But proteins, and maybe these regions are not as flexible. Proteins are kind of flexible.
Right.
Does that just work out in the neural networking that it learns how to deal with that? Or is that something you also have to take into account? And then I guess,
Yes, there are a couple ways to deal with that. So one is you can do ensembles where you take like a bunch of different representations of surfaces and then find something that kind of fits one of those or multiple of those. The other thing you can do is coarse grain it. So instead of looking at it at the like atom level, you can make kind of a blobbier, something that looks a little bit blobbier. So there are a lot of ways to deal with it. We like coarse graining because we can get a general idea of the shape, but we don't have to be super specific about that. But it also depends on the epitope of interest, because you could have epitopes that move around a lot more, where even at a coarse grained, something that's coarse grained, you're going to have a very big shift in the shape. And so in that case, you would do something like model 3 different systems and then design against all 3. And then, I mean, that's what the lab is there for, is to test which one actually did work. Often, the other thing is sometimes what you want to do is lock it into one of those shapes. And so that's actually a really big advantage of computational methods, because you can say, you know, you have these 3 potential shapes. We want to lock shape A. And so you make sure that it only works with shape A and not with B and C.
right. So kind of following up on that. So you're showing large proteins with epitopes. Does this also work if you're testing against a toxin, or it used to be transition state molecules? They were trying to get enzyme activity out of antibodies. Do those things work?
Honestly, like, I just don't work with small molecules, so I don't know. It's very different because the physics that drive those interactions are very different. So you can't coarse grain it out in the same way, because you lose a lot of that interaction. You really have to be a little bit more specific and more accurate in order to get at that.
Okay, great. This is fascinating.
Okay, Todd, and then we have a few more questions in the chat. I know, Monica, I'm not sure how much time you have, so please let me know if you need
Yeah,
to.
I could probably stay another like 5-10 past the hour.
Okay, so Todd.
Oh, excellent. Yeah, so before I ask the question, I was going to say, so the small molecule world, there's another molecule we call an aptamer, which is RNA or DNA, that its 3-dimensional structure can actually interact with small molecules very well. And so when you think about molecular recognition, there's a lot of ways to detect molecules. Yeah, my question is that, the explosion in computational biology also, the buzz, how do you see AlphaFold and things like that impacting your work, and pros and cons?
Yeah, I mean, so there are two basic ideas behind it. One is like, how does AlphaFold predicting more structures affect? And in that case, it's great, because one of the things that we can do that a lot of others can't do is we don't need a crystal structure, because we can rely on bad homology models. But obviously, the better your homology model, the better your final results. So being able to access like some of the AlphaFold structures just gives us so much more to work with. So in that case, I think it's a really big benefit. The other is, how can you adapt things like AlphaFold in order to deal with design? And I think that's one of those areas that like there's still a lot of room for that, because it's a very different way of thinking it. So one of the advantages of protein folding is you do not need a wet lab in order to verify that you're correct, right? So you can use crystal structures, you can train on crystal structures, you can train on data without ever having to go to the lab, and have a very good prediction of your accuracy without ever doing anything in the wet lab. Design is very different in that sense, because there is no metric that you can use computationally to get there. And so one of the things that like AlphaFold and other AI systems have done very well is take advantage of that lack of feedback that you need from the wet lab. And so I think where it starts to break down is where you try to apply that same thing without the correct data. And then you're making these predictions, say something works, and you're never going to know whether it does. And it's something that's affected small molecules a lot because design of small molecules is has relied on a lot more of the computational space without wet lab. And so once people start to test those wet lab things, a lot of that question breaks down.
Okay.
All right, thanks.
Questions from the chat. Angela was wondering, if you've created an antibody for an antigen, what do you do when the antigen mutates? And is there a way that computational biology can predict variants before they emerge?
Okay, so kind of two different questions. So predicting variants is on the antigen side, not on the antibody side. And that's not really my space of whether you can predict it. I've seen some amazing work and read some great articles that show how you can predict that. But again, that's not my area of expertise. But what you can do with variants is, this is something that a lot of people have worked on with flu, which is that a lot of the flu proteins have regions that are hypervariable, meaning that they mutate a lot, but then there are regions that don't mutate as much. And so what you can do to take advantage of computation is by focusing in on those regions that don't mutate. And so this really goes back to that question of how do you pick that functional epitope? And so if you can use information like that, find these regions that are less likely to mutate because they're super important to the function of the protein, you're more likely to get antibodies that work against that. You can use that, and I think that's how they've made a lot of vaccines that are more resistant to variants by making a vaccine around a region that doesn't mutate. You can do the opposite too and target those regions with antibodies.
She had one other question, which concerned the bioinformatics tools you use. And I know mostly you're using a lot of your own proprietary algorithms and software, but are there other kinds of bioinformatics tools that you use that in the public domain?
I mean, so what we do, we really rely a lot on just like sequence, either alignments or looking at differences. So we use things like BLAST and Hammer. Those are the primary ones we use that are not our own. And then we've built a lot of kind of taking that information and building it into a system that tells us something from that. So one is like getting the information from physics, analyzing and getting something you can actually do with out of that information. That's really where we've focused on our tools.
So are there other questions? I guess everybody just wants to say thank you now. Well, we really appreciate this, Monica. It's really exciting hearing about what you're doing. And yeah, I can see where it has a lot of
Thanks again for the invitation. I will say, so I'm happy to connect on LinkedIn with anyone if you want to reach out. I would suggest you tell me that you attended this. Otherwise, I tend to ignore LinkedIn invitations. But if you tell me that you attended this, I'd be happy to connect with you. And I'm always open to answering more questions.
Well, thanks. So this has been great. All right. So everybody's wondering what we're going to do next. So now we've got some time for you to go meet with your teams again, but don't just leave yet. Okay. don't take off yet. So before you do that, you might want to take, if you need to take a short break, not now, but in a couple minutes might be a good time. And then you're going to meet with your teams and work on your presentations for tomorrow. We also have a software demonstration that will take place at 12 o'clock Eastern, or I guess it's 3 o'clock Eastern, 12 o'clock Pacific, and I will put that information into the chat. And after that, you'll have a little bit more time to work with your teams and get your presentations ready for tomorrow. Because tomorrow, each team is going to do like a less than 15-minute presentation on the work you're going to be tackling this week. Okay, so before you leave for breaks and your teams, are there any questions? All right, then. I will see some of you in breakout rooms. And remember, there will be a software demo at the top of the hour, or in about an hour. Okay. See you all soon.